Hi everyone, I'm Jana Eissa, and in this video we're tackling Chapter 18 Biochemistry, all about transcription. This video is question-based. We'll go through 15 MCQs that cover key aspects of the chapter. Ready to test your biochem knowledge? Let's go! Before we start, after every question, feel free to pause the video and then resume once you got the answer. Why is DNA more chemically stable than RNA? Have a look at the diagram. Let's compare the sugars in DNA and RNA. DNA contains deoxyribose, while RNA contains ribose. The key difference is, look at carbon number 2, and then look at what is attached to carbon number 2 and the deoxyribose and the ribose. RNA has a hydroxyl group, whereas DNA just has a hydrogen on the second carbon. So this hydroxyl group in the RNA makes it more prone to hydrolysis, and so if it's more prone to hydrolysis, then the answer is C. Especially, by the way, in basic conditions, that's when the hydroxyl group is more prone to hydrolysis. So RNA is inherently less stable because of this hydroxyl group, as it can participate in intramolecular reactions that cleave the phosphate backbone. Therefore, DNA's lack of this group makes it more stable, which is why we use DNA for long-term genetic storage. So I hope this concept is now clear. Which direction is RNA synthesized by RNA polymerase? Is it 5' to 3' or 3' to 5'? Now let me give you a hint. It's like DNA polymerase. I hope you got the answer now. RNA polymerase can only add, like DNA polymerase, nucleotides to the 3' hydroxyl group of the growing RNA chain. So in that case, it's going to be 5' to 3'. RNA synthesis always occurs in that direction. The enzyme moves along the template strand and assembles the RNA strand by forming phosphodiester bonds between the 3-hydroxyl of the last nucleotide and 5-phosphate of the incoming ribonucleotide. So again, what is the direction? From 5' to 3', but always adding a new nucleotide at the 3 end. Okay, what is the role of the sigma factor, not sigma point, in prokaryotic transcription? And really, think about the sigma factor as the GPS of the RNA polymerase. It helps the enzyme locate the right address. So the answer is C. It guides the RNA polymerase to the promoter. The sigma factor enables the RNA polymerase to recognize and bind to specific promoter sequences. And there are 2 specific promoter sequences that the sigma factor enables the RNA polymerase to bind to. One being the Pribnow box, which is 10 regions upstream, and also 35 regions upstream. That's where particularly the sigma factor is going to guide the RNA polymerase to bind to these 2 regions. Once transcription begins, so RNA polymerase has started transcription from the 5' to 3' direction, a sigma factor is then going to be released. Okay? Now, what is this Pribnow box that we just said that is a region at 10 nucleotides upstream, and where is it going to be located? So part of the answer is already been told, so I guess you can already say that it's C. The sequence plays a key role in transcription initiation in prokaryotes by facilitating the unwinding of the DNA, because we need the DNA to unwind in order to allow for transcription to occur. So we need this region for the initiation of transcription. So the Pribnow box is a region in bacterial promoters, centers approximately 10 base pairs upstream of the transcription start site, and it is rich in adenine and thymine pairs, making it easier to unwind due to weaker hydrogen bonding. So the answer is, and therefore, C. Now, what happens to the sigma factor after initiation of transcription? After its job in helping, and I've already mentioned this, by the way, so I think you've already gotten the answer, but after its job in helping the polymerase find the promoter is done, the sigma is no longer actually needed, or the sigma factor is no longer needed. So the answer is C. It's released from the RNA polymerase. So once RNA polymerase has synthesized about 10 nucleotides of RNA and cleared the promoter, the sigma factor dissociates, and this allows the enzyme to continue elongation more efficiently. And that's why the answer is C over here. Okay, which statement best describes the transcription bubble? So have a look at the diagram. Have a look what is happening to the 2 DNA strands. Now, the transcription bubble is essential for RNA polymerase to access the template strands. You can see that they're separated from each other. So the answer is B, a small open region of unwound DNA where transcription occurs. A transcription bubble is typically 12 to 14 base pairs long and contains a short RNA-DNA hybrid. It moves along the DNA as RNA polymerase progresses, temporarily separating the DNA strands. So without this transcription bubble, the DNA itself is not going to be accessible, and therefore transcription is not going to occur. So that's why the transcription bubble is very, very important to be able to make the DNA accessible for transcription. Okay, moving on. What differentiates row-dependent from intrinsic termination in prokaryotes? We're specifically focusing on these two. We've now gone over the initiation, we've gone over the process of transcription, the theory behind it, and now we've reached the termination part in prokaryotic transcription. So what is the difference between them? These two, by the way, they're distinct mechanisms that stop transcription specifically for prokaryotes. The answer is B, where dependent uses ATP dependent helicase. Then which of these statements do you think applies for the intrinsic termination? C would be for intrinsic termination. If I said intrinsic termination relies on a hairpin loop, in that case, C would have also been a correct statement. So row-dependent termination, the row protein, the ATP dependent helicase binds to the nosin RNA and unwinds the RNA-DNA hybrid at the termination site, releasing the transcript. The intrinsic termination, on the other hand, relies on a hairpin loop and a uracil-rich region without protein assistance. So whenever you think about row-dependent, think about ATP, dependent helicase, and then whenever you think about whenever you think about intrinsic termination, think about hairpin loop and uracil-rich regions. So that's the difference between the two possible terminations that we can have for prokaryotes. So now we're done with prokaryotes. Now we can go for transcription in eukaryotes. Which RNA polymerase synthesizes mRNA in eukaryotes? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And it's RNA polymerase 2. RNA polymerase 2 is responsible for transcribing mRNA precursors, miRNAs, and most snRNAs. Basically, RNA polymerase 2 very, very important when we come to talk about eukaryotes and transcription that happens in eukaryotes. Okay. Now, what is the first RNA modification after transcription initiation? So I've started transcription, and I'm already going to start modification. It's not like I'm going to have to finish transcription first and then I start modification. There's something that happens as soon as I start transcription initiation in order to protect the sequence that I'm building. The answer is B. And this modification, as I said, protects the RNA and aids in ribosome recognition. The 7-methylguanosine cap is going to be added to the 5' end of the transcript shortly after the initiation, after, say, approximately adding 13 nucleotides. This cap protects the mRNA from exonucleases. Now, exonucleases are essentially enzymes that cut at the end of a chain. Endonucleases are those that are going to cut in the middle of a chain, okay? And it helps initiate, therefore, translation. So, therefore, 5-capping is the first modification that's going to occur soon after the initiation of transcription in eukaryotes. Now, what sequences splice introns? Now, remember, the mature mRNA should only have exons, not introns. In eukaryotic pre-mRNA, is it an AUG codon? AUG is start codon, so no. Is it the Shine-Delagrano sequences? No. Poly A tail? That's not related to the splicing process. So C is the right answer. The Shine, by the way, sequence, this is the one that we find mainly in prokaryotes. So this is not relevant in splicing here, splicing of introns. So it's conserved from the 5', the guanine, uracil, and then the 3', we've got adenine, guanine. That's going to be at the 3' end. So most eukaryotic introns begin with a guanine uracil and end with an adenine guanine, and these conserved motifs are recognized by components of the spliceosome to accurately define the intron boundaries. So this is going to be the signal This part over here is an intron, and it has to be removed to produce a mature mRNA. Now, which RNA polymerase synthesizes 5S rRNA and tRNAs? It's more of a memorization one. It's either right or wrong. So the answer is C, RNA polymerase 3. So RNA polymerase 3 is the one that's going to transcribe genes for some sRNAs, tRNAs, 5S rRNAs. So don't confuse that with the RNA polymerase 2, which is essential for mRNA production or synthesis. Now, which of the following is not a function of the 5' cap? We already touched upon this. The answer would be spliceosome recognition. We don't need that for spliceosome recognition. We said for spliceosome recognition, mainly we need to recognize the introns, and the introns we said they start with guanine, uracil, end with adenine and guanine. So the 5' plays an essential role in RNA stability, nuclear exports, and also translation initiation, but it's not directly involved in spliceosome recognition, which depends on, as we said, the intronic and exonic sequences. So that's why the answer is D, because it's not a function of the 5' cap. Now, what does polyadenylation do? Polyadenylation. So that means I'm adding a a tail. Answer is C. It adds a tail of adenines to stabilize mRNA. Polyadenylation involves adding a poly-A tail to the 3' end of the mRNA after cleavage. It enhances mRNA stability and regulates translation efficiency. So that's why the answer is C. It's part of also the modifications that a pre-mRNA has to undergo in order to become a mature mRNA. Now, which sequence directs then the polyadenylation signal? The production of this poly-A tail, or the addition of the poly-A tail to the maturing mRNA. Answer is C. So the adenine, uracil, adenine, hexamer. So this sequence is recognized by cleavage and polyadenylation specificity factor, also known as CPSF. Cleavage and polyadenylation specificity factors, and it defines the site of cleavage for the poly-A tail addition. Okay, so this is why it's very important sequence, again, for this modification process for the addition of the poly-A tail. Now, going on to the last question, what happens when the C-terminal domain, also known as the CTD, of the RNA polymerase 2 is phosphorylated? So we're going to phosphorylate the CTD on the RNA polymerase. So phosphorylation here acts like a switch for downstream processing. The answer is C. Transcription and RNA processing begins. It's not that polymerase is deactivated. It doesn't deactivate polymerase. Transcription does not stop. It continues, and actually it helps with the RNA processing, and there's no disassociation from the DNA. So the answer is C. The CTD of the RNA polymerase 2 contains something known as the heptapeptide repeats, and then phosphorylation of specific serines allows the polymerase to initiate elongation of the mRNA and to recruit capping, splicing, polyadenylation, and all of these processing for the multiplication of the mRNA to produce a mature mRNA. That's it. I hope you found this video useful and the questions useful. I hope that I was able to cover the most important aspects of Chapter 18. If you have any questions, please feel free to ask me any questions. That's it for Chapter 18. I hope you feel more confident with transcription now, and I hope you have a wonderful day. Bye-bye.
Alright, so hello everyone, my name is Assia, and today I'll be presenting Chapter 19 of Biochem 2, which is protein synthesis. So I hope this will be beneficial to you guys. Alright, so the two parts included in this chapter are the genetic code and protein synthesis. So we'll start with the genetic code. So protein synthesis is also called translation because we are translating a sequence of nucleic acids to a sequence of amino acids. So the nucleic acids are read in codons, which are sets of 3 nucleic acids. So we have 4 nucleic acids and sets of 3. So 4 to the power 3 gives us 64 different codons possible. However, we only have around 20 amino acids, which means that many amino acids will be repeated. And in addition to this, we have 3 stop codons that do not code for any amino acid. All right, so here we have the genetic code. So we can see that each codon corresponds to a single amino acid. And we have 4 very important codons to memorize. So we have the start codon, AUG, which codes for methionine, and the stop codons, UAA, UAG, and UGA, which will stop the translation process when the peptide is finished. So these 4 are to be memorized. They're very important, and you will get questions on those. Next, we have the 4 properties of the genetic code. So the genetic code is specific. So as we've said, each codon codes for a single amino acid. So we will not have the same codon giving you different amino acids. It's always the same. However, the opposite is not true, because the genetic code is degenerate, which means that one amino acid can be coded by multiple codons. So we've mentioned only 20 amino acids, but 64 codons. So many codons will give the same amino acids. The third property is non-overlapping. So these codons are successive. So we're going to read 3, then 3, then 3. We won't read 3, then come back to the second and read from that, and then come back from the third and read from that. No, we're going to read 3 of them, then forget about them and move on to the next 3. So the nucleic acids are not repeated in the codon sequence. And finally, the genetic code is universal, so it is nearly always the same, whether it is in a human cell, in a bacterial cell, in a fish cell, whatever you want. It's nearly the same wherever you go, with slight differences, but nearly the same. All right, so now we move on to the role of tRNA. So tRNA, or transfer RNA, will match each codon to a corresponding anticodon. And on the other end of the tRNA, we will have the amino acid that corresponds to the codon. So at the end, it matches the codon with the corresponding amino acid, and it does that through an anticodon. So for this process to happen, we have a synthetase reaction, which is the process of linking a specific amino acid to a transfer RNA. It is catalyzed by amino acid tRNA synthetase, and it is a highly accurate process, despite how complicated it is. And this is because the amino acid tRNA synthase sometimes has proofreading sites, which allows it to correct certain mistakes it might have done. So we can see in this figure that the synthetase reaction occurs in 2 steps. So first we have the amino acid combined with ATP, and then from there it will lose the ATP and instead combine with tRNA. All right, so here we have a question for you guys. So which one of the following is true about the genetic code? So I'll let you guys read the answers. You can pause the video to answer. And in a few seconds, I'll be giving the answer. All right, so let's go by elimination. First, all codons recognized by a given tRNA encode different amino acids. Is this true? No, because we've mentioned that a single codon, a single amino acid can be coded by different codons. As we mentioned, since the genetic code is degenerate, multiple codons can code for the same amino acid. Now, is it absolutely identical in all living things? No. Why? It is universal, but not 100%. It is nearly always the same, not always the same. So, option C, several different codons may encode the same amino acid. Is this correct? Yes, it is correct. This is what I've mentioned about the degenerate nature of the genetic code. Now, the first position of the tRNA anticodon is always adenosine. Is this correct? No. Why? Because we know that all translations start with the same codon, which is AUG. So the anticodon corresponding to AUG would be UAC. So every first tRNA anticodon will start with U, not A. So uracil, not adenosine. All right, so moving on to protein synthesis. So there are 3 steps in protein synthesis. We have initiation, elongation, and termination. So initiation, as we've mentioned with methionine, then elongation with a peptide bond formation from amino acid to amino acid, and then termination with a stop codon that will induce hydration, which stops the peptide from growing. So to start with, we have initiation. So first, the tRNA will bind the AUG on the mRNA. Next, we'll have the ribosome that intervenes. So the ribosome is the protein that is in charge of translation. So it is made up of 2 subunits, a small and a large one. So first, the small subunit will bind to mRNA, and then the large subunit will bind to tRNA. All right. So multiple ribosomes can read the same mRNA at once, and this compound of a single mRNA with multiple ribosomes has a name. It's called a polysome. So for the second picture I've put here, we have a ribosome with 3 sites. So these 3 sites are important because each tRNA will go from one site to the next to the next. So it first comes to the A site, then moves to the P site, the peptide bond will be formed, and then when it moves to the E site, it is ejected from the ribosome. So remember E ejected, so you know that the E site is the last site by which the tRNA will be passing. All right, next we have elongation. So the mRNA is read in a 5 to 3 direction, and the protein is assembled from an N to C from the N terminus to the C terminus. So these orders are very important because, as you might remember, for DNA replication, it's going to be a certain order. For mRNA formation, it's going to be a certain order. And for translation, it's also a certain order. And it is easy to get mixed up in these. So make sure to keep all of this in mind so you don't get tricked in the exam. So there are also 3 steps in elongation. So first we have the codon-anticodon pairing in the A site. Then we will have a peptide bond with the previous amino acid, which is now in the P site. And then this tRNA will move to the P site, and the one that was in the P site will move to the E site, as we can see in the illustration here, in this figure. All right, next we have termination. So the stop codon will bind a protein releasing factor. So the stop codon, instead of binding to tRNA, will bind a protein releasing factor, and this factor will trigger an enzyme called peptidyltransferase to cleave the bond between the growing peptide and the last tRNA. And then finally, the ribosome releases the mRNA and dissociates into its subunits, and we end up with a new peptide. All right, so now we move on to prokaryotic protein synthesis. So there are slight differences between protein synthesis in prokaryotes and in eukaryotes. So you guys might remember prokaryotes are specific beings with a certain type of cell. So human beings, for example, are prokaryotic, while eukaryotics would be more like bacteria. So for prokaryotes, the ribosomes are made of a 50S large subunit and a 30S small subunit. And we go into even more detail as the 50S is going to is further divided into a 23S and a 5S rRNA, as well as 34 proteins, while the smaller subunit has 16S rRNA and 21 proteins also. So you might get questions on these, so remember this well. Next, we have the peptidyltransferase, which is located in 23S. So you guys remember right before peptidyltransferase is the enzyme that cleaves the bond between the peptide and the last cRNA. So this enzyme is found in the large ribosome subunits, and specifically in the 23S rRNA. So remember this if you get asked about the location of peptidyltransferase, for example. Then we'll have some post-translational modifications in these proteins, and these will be triggered by trigger factors. So these post-translational modifications, the most common would be the removal of signal peptides and of formylmethionine residue. So we remember that every peptide starts with methionine. However, not every functional protein in the world starts with methionine. So between the production and the functioning, this methionine residue has been removed. Now for signal peptides are peptides that bring the peptide from non-functional to functional. So here we have the example of insulin. Oops, all right. Insulin starts as pre-proinsulin with a signal peptide. Then once the signal peptide is removed, it becomes proinsulin, and then finally it becomes insulin, which is the functional molecule. Now for eukaryotic protein synthesis. So there are a few differences. For example, the initiation is going to be more complex. So how so? The mRNA is going to be more processed even before starting the translation, and the translation is helped by other proteins. So it's not only going to be ribosomes and RNA. And in addition to this, ribosomes will scan for the translation start without the help of a shine-dog RNA sequence. So a shine-dog RNA sequence is a sequence that is found in prokaryotes and that will indicate the start of the translation. However, eukaryotes do not have this sequence. Instead, eukaryotes have at least 12 different eukaryotic initiating factors, which causes the entire process to be a little bit more complicated. And then finally, the protein will undergo over 200 different post-translational modifications. And just as a side notice, post-translational modifications are covalent, so they're here to stay. All right, so here we have the 8 main types of post-translational modifications. So we have proteolytic cleavage, glycosylation, hydroxylation, phosphorylation, lipophilic modifications, methylation, carboxylation, and disulfide bond formation. So starting with proteolytic cleavage, so we've mentioned earlier, it's the removal of the methionine or of signal peptides to activate a protein. Then we have glycosylation, which is adding a carbohydrate to a protein, and it may have a role in the function of the protein, but it may also just serve to protect the protein from degradation from the endoplasmic reticulum. Next, we have hydroxylation. So this only happens to two amino acids, proline and lysine. So you guys might remember from the protein chapter that we have hydroxyproline and hydroxylysine in collagen. So hydroxylation is an important reaction in the process of formation of collagen. It's also included in the formation of acetylcholinesterase. And hydroxylation requires vitamin C, which explains the disease scurvy. As you guys might know, scurvy is due to a deficiency of vitamin C. Your old scars will reopen because there is no more collagen to hold them closed. So here's why. Then we have phosphorylation, which plays a role in metabolism, control, and signal transduction, among us. We have lipophilic modifications, so adding a lipid to a peptide. And as we know, plasma membranes are lipophilic, so adding a lipophilic modification will improve membrane binding capacity of these proteins. And two examples of lipophilic modifications are acylation and prenylation. Next, we have methylation. So it marks proteins for repair or degradation. And we have carboxylation, which only happens to glutamyl residues and is involved in blood clotting. This process requires vitamin K. And you guys might know from the blood chapter in Bio 2 that certain clotting factors require vitamin K to be synthesized. So this is why a deficiency in vitamin K will cause problems in blood clotting. And finally, we have disulfide bond formation. So we have we can find this in secretory and membrane proteins, and this bond is mostly formed in the rough ER because of the non-reducing environment of the rough ER. All right, and now for some questions for you guys. So the large structure consisting of an mRNA molecule being translated by multiple copies of the macromolecular complexes that carry out protein synthesis is called a... I'll leave you a few seconds. You can pause the video, think about it. All right, so let's start by understanding the question. We're saying multiple copies of macromolecular complexes that carry out protein synthesis. So this entire expression refers to ribosomes. So the question is asking, how do you call one mRNA molecule being translated by multiple ribosomes? So this is the definition of a polysome. So the answer is B. All right, so next, which one of the following statements about ribosomes is true? So again, a few seconds to think. All right, so let's proceed by elimination. The large subunit contains rRNA molecules, the small subunit does not. This is incorrect. You can go back to the smaller subunit, it also contains rRNA. The RNA in ribosomes plays a structural, not catalytic role. This is also wrong. Remember the protein transferase that we mentioned, the enzyme that was in the larger subunit? It was also an RNA, and as an enzyme, it plays a catalytic role. There are about 25 of them in an E. coli cell. This is not correct. There are over thousands of ribosomes in each cell, whether it's an eukaryotic cell or a prokaryotic cell. There are two major subunits, each with multiple proteins. Is this correct? Yes, it is. It is. They are relatively small with molecular weights less than 10,000. This is incorrect. They're very large molecules. They themselves contain proteins, contain RNA, so obviously they're very large. All right, and here is a little crossword for you guys. So you might want to try it. have a go at it. You might find this one. And that's it. That's all I have to say. So I hope you guys benefited, and good luck in your exams.